the spinning ball. There we go. All right, let us go ahead and, as they say, kick this. Right. Okay, window, present, there. Make sure I've got the right one. Right. So, what's plankton? Aside from that little thing shaped like a vitamin pill that does something in SpongeBob. Sorry, my kid was never much into SpongeBob, and frankly, neither was I. Plankton are organisms that live in the water column, meaning they live above the bottom, and that can't swim strongly enough to fight typical currents. Some may not swim at all, others may have limited ability. Many of them are capable of rising and sinking. In fact, a lot of planktonic organisms do a daily cycle of sinking by day and rising by night, called BIOL migrations. We'll talk more about those later. But plankton can't generally swim strongly enough that they can fight typical currents. Jellyfish, for example, can swim, but if the current's flowing strongly, they pretty much have to go with it. And we can talk about plankton being either phytoplankton or zooplankton. Phytoplankton are photosynthetic. Phytos means having to do with the plant. And so these are the plant-like, sunlight-absorbing photosynthetic bacteria and some protists, things we usually call algae. Zooplankton are heterotrophic, meaning they eat other things. That would include some protists and also, of course, animals like jellyfish. A second way of dividing plankton is dividing them depending on how much time or how much of their lives they spend as planktonic organisms. Holoplankton spend their entire lives as plankton, spend their entire lives floating. They may never contact a solid surface in their lives. Meroplankton will spend part of their lives as plankton, but the other part somewhere else. Jellyfish, for example, many of them actually have this alternation of generations life cycle where they'll spend part of their lives as a little polyp, a little blob on the ocean floor, and that will bud off jellyfish, which will reproduce sexually and produce larvae that grow into blobs on the ocean floor. So the life cycle actually switches between a stage on the floor and a stage in the plankton. A lot of animals have larvae that do that, as we discussed on Friday. An awful lot of marine snails and echinoderms, that's starfish and sand dollars and all that, are meroplanktonic because they'll spend part of their life cycle in the plankton and the other part probably on the bottom. We can also talk about other subdivisions of plankton. This is Yanthina. That's actually a snail with a rather attractive purple shell, I might add, that sometimes is found washed up on beaches. Yanthina is actually able to secrete gas bubbles and strengthen them with mucus. And so it blows this more or less permanent bubble raft and hangs onto that and spends its entire life floating. And because it spends its entire life not just floating, but right at the air-water interface, right at the surface tension, we call that a member of the neuston. So neuston is specifically plankton that's right at the air-water interface. And I should mention that there was a Yanthina snail that actually became very famous because these snails are capable of making sounds. And this Yanthina snail was actually able to sing. I'm sure you've all heard of Whitney Houston. Okay, I'm envisioning you all groaning right there. Subtype of Neuston is Plouston. So this is a Plouston-Neuston plankton right here. These are colonies of, we call them hydrozoans. They are, let's just say, kin to jellyfish. In fact, most of them do alternate between a jellyfish and a polyp stage. They grow, almost all of them, in colonies. Most of the time, the colonies grow on the bottom, but what you're looking at here is a colony of little blobs with tentacles on it that is so tightly integrated that it looks like a single organism. This is actually a colony of polyps, each with a mouth and tentacles, all of them feeding. Some of them specialize for reproduction. And when the colony gets together, they can actually secrete an internal structure that is filled with gas, and it's divided into chambers. It's actually kind of like a pool floaty. And even better, there's a fin extending up from this round pool floaty thing, and the fin can actually catch the wind. So Plouston not only live at the air-water interface, but they spend, they've got a significant part of their bodies actually literally sticking up into the air. So this colonial organism that you're seeing is called Velella, common name sailors by the wind. These wash ashore in great numbers sometimes. I used to see them washing ashore in drifts on beaches in the San Francisco area in Northern California. You would see part of the ocean covered with this beautiful blue but slightly slimy stuff, and it turned out it was masses of these Velella, each of which is roughly an inch across, maybe a little more. So that's a beautiful example of organisms that are Plouston, Neuston plankton. So there we go. Sorry, I keep imagining Velella singing, and I will always love you. Ooh. Still got the high notes. Right. And of course, we can also talk about planktonic organisms based on, you know, the biological groups they belong to and what they do. One of the most common planktonic organisms are bacteria. There's bacteria everywhere, of course, but there's plenty that live in the oceans. Pelagibacter ubique feeds on dead organic matter, and it turns out that this makes up 25% of all microbial plankton cells, sometimes as much as 50%. Ubique happens to be Latin for everywhere. So this is the planktonic bacterium that is everywhere. And it's also got the smallest genome known of any free-living organism. 1.3 million base pairs. That's something like 1/3000th the size of yours. You've got something like 3 billion. Only 1,354 protein coding genes. There are living things with smaller genomes, but as far as I know, they're all parasites. Pelagibacter has the most stripped down genome of any organism that lives on its own, and it's incredibly common in the oceans. It's everywhere. So is, this is a phytoplankton organism. You're looking at a cross-section through it, imaged with a transmission electron microscope. It's called Prochlorococcus. That's a pair of cells in the process of division. Each cell is less than 1 micron in diameter. And it turns out it's the single most abundant photosynthetic organism in the world. There may be as many as 100,000 cells per milliliter in seawater at the surface. And you might never see it. Even under a light microscope, you would be hard-pressed to spot it. Light microscopes have a hard time seeing details smaller than roughly 1 micron. But Prochlorococcus is extremely abundant, and before 1986, we didn't even know it was there. Take you back in time to the late 19th century. There's a guy by the name of Sir William Thomson, who is probably better known by the name that he got as part of his title of nobility from Queen Victoria, known as Lord Kelvin. Kelvin was a great scientist. You know, he got a temperature scale named after him, and there's not too many people that have, you know, he's one of 3, you know, beside, you know, Mr. Celsius and Mr. Fahrenheit. You know, he worked out an enormous amount of the physics of heat transfer and thermodynamics. As he got old, he started making some pronouncements that, well, he's the guy who claimed that heavier than air flying machines were impossible, that radioactivity was a hoax, X-rays were a hoax, and radio had no future. He tended to get a bit stuck in his ways and maybe a little bit, you know, let's just be kind and say a little bit of a stick in the mud by the end of his life. And he did some math, calculated in 1898 that at the rate at which wood and coal and things were being burned, the Earth was going to run out of oxygen in 500 years or less. So he was able to calculate the rate at which oxygen was put into the atmosphere by plants, because you could estimate, you know, how many land plants there are. You can measure how much oxygen they put out, and then you can measure the rate of oxygen consumption, estimating the amount of stuff that's burned every year. And he did these calculations and calculated that in the course of 500 years, there will not be enough of the gas left on the Earth to support life. However, he did the math right, but his assumption was that the only source of oxygen was land plants. We now know that, well, of course, there are plenty of photosynthetic organisms in the ocean. About 20% of global photosynthesis is carried out by Prochlorococcus cells. And even though each one is small, there's an estimated 10 to the power 27 Prochlorococcus on Earth at any given moment. Yeah, this is an organism whose abundance you could measure in moles. So this is what, something on the order of, what, 1,000 moles of Prochlorococcus cells on the planet. And by itself, Prochlorococcus puts about 20% of the Earth's oxygen into the atmosphere. So if you're breathing, thank Prochlorococcus. And again, we didn't even know it existed until 1986. There's even planktonic viruses, and these are pictures taken through a transmission electron microscope of 4 of them. These are called cyanophage viruses because they infect Prochlorococcus and other planktonic bacteria. Nobody really knows anything about these guys. We know they're there, and they're probably important in regulating bacterial populations, but haven't been studied very much. Something for you to think about if you're thinking about going on to a career in research, maybe. Okay, so that's bacteria and viruses. Let's look at the protists now. Protist being a catch-all term for any eukaryote that isn't an animal, plant, or fungus. If you've taken or will take invertebrate zoology, you know we divide those up a bit differently, but I'm going to stick with protist here just for simplicity. Many protists are multicellular. Many of them are also unicellular. These 3 groups that I'm going to talk about are single-celled or maybe form very small colonies. And the three big groups, and you need to know all three, are diatoms, dinoflagellates, and coccolithophorids. Diatoms you could actually see in freshwater samples. A small smear of mud from Lake Conway, and you'd see them by the dozen under a microscope. There's lots of them in freshwaters. And diatoms are photosynthetic. They have chloroplasts that contain kind of yellow and brown pigments in addition to chlorophyll. So living ones often look kind of golden brown. They don't look obviously green. The cells are very symmetrical because the cells are encased in cell walls that are impregnated with silica. Diatoms are literally made of glass. And they can grow and they can divide because the cell walls are composed of a pair of valves that fit together like the two halves of a petri dish, which I assume you have seen before. They're these round dishes with a lower half and then an upper lid that fits right over the lower half. That's a passably good model of what a diatom frustule, the silica cell wall, looks like. These two valves that fit together. And that enables the diatoms to reproduce. They can divide, and then each cell takes one of the valves and builds a new one. Dinoflagellates you could also see in Lake Conway. There's lots of these in freshwater. They swim, and they spin as they swim. Dino, in this case, comes from a Greek word meaning whirling or spinning, not the same as the Greek word meaning frightening, right? Dinosaur means frightening lizard or terrible lizard, with terrible in its old sense of scary. This dino means spinning. These are the spinning flagellates. They're eukaryotes. They've got a cell wall made of cellulose and two flagella with which they swim. And then the third group that you won't see in freshwater, called coccolithophorids, they're photosynthetic, they're small, and they're covered with little plates of calcium carbonate called coccoliths. Phorid just means it carries. So coccolithophorid means things that carry coccoliths. So let's see what these look like. Here are some marine diatoms. Some of them are extremely elongated. We call such diatoms pennate. If they can be anywhere from extremely elongated to oval, to football-shaped, to even S-shaped or zigzaggy. And then radial diatoms are circular, or sometimes they're very regular triangles. I can see one of those in here, two of them, or occasionally other shapes. So that's what they look like. Again, that golden color is because they're photosynthetic, but they add yellow and brown pigments as well as the chlorophyll, so they don't look obviously green. And the valves are ornamented with ridges, perforated with little pores and little slits, and often they are just beautifully symmetric. So you can see a valve right here. It's a little bit broken, but you can see it's got these very delicate ridges and pores, and the pores are, maybe half a micron in size. You could barely see this detail with a light microscope. So yeah, diatoms are gorgeous, very symmetrical like that. Dinoflagellates, just under the plasma membrane, have a bunch of plates made of cellulose that fit together to make a firm covering called a theca. And this is a theca. You'll notice the cell itself is kind of diamond-shaped, and there's a groove running around the midline of the cell, like a belt. That's called the girdle, because it looks like a belt or a girdle. And if this thing were alive, one flagellum, one of these long hair-like organelles beating back and forth, would be trailing along behind it and beating to push it forward, and the other would be wrapping around the girdle and beating to cause it to spin. That's just the way they do things. There are a few that are naked. These are some Noctiluca that don't have that cellulose armor. These get pretty large, up to 1 millimeter across, although most of the cell is empty. It's mostly just a big vacuole. But they have flagella, and some dinoflagellates also have a little feeding tentacle. This particular one is not photosynthetic, although many dinoflagellates are. Some aren't. Some of them are carnivores. When you get a big bloom of dinoflagellates in a body of water, you get something like this. That's a picture of Florida a few years back. Notice there's a bunch of dead fish in the water, and the water looks like it's turned to blood. That's not the prophecy of the book of Revelations being fulfilled, at least not in this case. That's a red tide. That's a bloom of a particular dinoflagellate, huge numbers of it. The fish are dead because many dinoflagellates produce toxins, especially in quantity. If you have a bloom, many of them will produce toxins. And if fish, or especially shellfish, oysters, clams, things like that, if they filter feed on dinoflagellates, they can build up toxin in their own tissues. And then if you eat the shellfish, those toxins can cause very severe illness. There's a condition you can get called paralytic shellfish poisoning, caused by eating, clams and oysters and things like that. It's not actually the shellfish that are causing that. It's the fact that the shellfish have been filter feeding on so many dinoflagellates that they've built up large amounts of dinoflagellate toxin. There's a similar disease that can have effects on your nervous system called ciguatera that you can also get from this. One dinoflagellate in particular, Pfiesteria, allegedly is so toxic that a couple of scientists that were growing it in the laboratory for study spilled some on them, and allegedly they've still got things like memory problems and physical symptoms. I say allegedly because I'm not entirely sure. There was some debate as to exactly what was causing it, but certainly claims have been made that some of these things are extremely dangerous. Red tides can be caused by inputs of fertilizer. One concern is that these have become more common in places like North Carolina at the same time that hog farming has taken off, and we've had spills of hog waste into rivers that flow into the ocean. You dump enough fertilizer into the ocean and you'll get things growing to feed on it, and the fear is that sometimes that's dinoflagellates. Never let it be said that they're all bad, though. There are some places in the world where there just happen to be more or less permanent large populations of dinoflagellates. And one of them is just off the island of Vieques, which is next to Puerto Rico. Vieques, you used to not be allowed to visit because it was a military bombing range. It's still dangerous to go on to Vieques. There's a lot of unexploded bombs that they're going to have to clear. They stopped bombing a few years ago, but it's going to take a lot longer to clear away the unexploded ordnance and make it safe to actually go on the island. But now you can take kayak tours to a bay on the island of Vieques that just happens to have a very high population of a dinoflagellate called Pyridinium bahamense. The cool thing about Pyridinium bahamense is if you disturb it, it starts screening light. It's bioluminescent. And you can see these people in their sea kayak where their oars are dipping into the water. You can see that there's this eerie blue light. There's a whole trail of blue light behind them where their canoe has been. That's Pyridinium bahamense. The thinking as to why they might light up is a little analogous to a car alarm. The idea is that if a predator comes and starts eating them, if they all light up, then all of a sudden the predator has drawn attention to itself, and maybe critters that would eat the predator are going, hey, wow, what's that light? Hey, look over there. So the thinking is that a mass bioluminescence response might be an anti-predator mechanism. At least that was the thinking a couple of years ago. So there, maybe someday you can go to Vieques. You might never get to swim with the dolphins or swim with the whale sharks, but at least you can go kayaking with the dinoflagellates. So there. Third major group of photosynthetic protists are coccolithophorids. They're a lot smaller than the others. These cells are maybe 5 microns across, whereas typical dinoflagellates and diatoms could be 10 times that. The cells themselves are small and round. They've got flagellum, so they swim. Internally, though, the cells secrete these plates of calcium carbonate and export them to the outside. So these are covered with these very geometric calcium carbonate plates. The plates are called coccoliths. Kokkos means a little round thing, and litho is Greek for stone. So these are little round stone things. And this is how abundant they can be. That's a satellite picture of the southwestern tip of Great Britain. Most of this is the part of Great Britain known as Cornwall. In the lower and the lower right part of this picture, that's showing the English Channel. So just off of the edge of the picture would be France. And within the English Channel, you can see there's this kind of eerie blue milky stuff. That's coccolithophorids. The water is turned turquoise, and they may extend for hundreds of miles. The width across of this picture is very roughly 50 miles or something like that. And you can see that, from one end of it to the other, you can see this giant bloom of coccolithophorids. So that's even bigger than the red tide that we saw. These are not particularly harmful. Coccolithophorids don't hurt anybody as far as I know. What they'll do is live and photosynthesize and reproduce and die, as all things must, and then sink to the bottom. Those plates of calcium carbonate don't go anywhere. They're not going to decay or anything. So they'll build up on the ocean floor. And this went on in the past as well, something on the order of 70-ish million years ago, and created this. That's the White Cliffs of Dover on the southern coast of England. These days, I guess most people take the tunnel under the English Channel. But when I was there, you had to cross by ferry. And the White Cliffs of Dover was, first thing that you saw as you were leaving France or Belgium and crossing the English Channel to Dover, the main port for cross-channel traffic. And these white cliffs are millions of years old. They're made of natural chalk. Chalk is basically a very soft limestone. And most of this rock is fossil coccoliths. So you've got a layer of fossil coccoliths that extends for several miles, and at its thickest is, I'm not sure, 100 feet thick or something like that. And it's made up almost entirely of coccoliths. That should give you some idea as to how abundant these things were in the past. And in fact, in some parts of the ocean, they can still be that abundant. It's not as impressive as Dover, but if you were to drive down to, the general metropolitan area of Saratoga, Arkansas, and poke around a little bit and know where to look, you could find a rock layer that is tops maybe 20 feet thick and extends all the way to Texas, called the Annona Chalk, which is also made up mostly of coccoliths. I've actually collected some down there. So they're abundant enough that they can build up sizable layers of sediment on the ocean floor. And if those harden into rock, you get things like the White Cliffs of Dover, or the crumbly white rock exposed just outside of Saratoga, Arkansas. Right. So those are the main players of phytoplankton. Bacteria, blue-green bacteria, and of course, dinoflagellates, diatoms, and coccolithophorids. What's eating them? Plenty of animals, and we'll talk about planktonic animals on Wednesday. The protists that are feeding on all of these guys, on these bacteria and blue-green algae and so on, also belong to three primary groups: tintinnids, foraminifera, and radiolaria. Tintinnids are ciliates, and in previous classes, you've probably at least seen a ciliate called paramecium. Looked a little bit like a slipper covered in little beating hairs, pretty large as eukaryotic cells go. If any of you have taken cell biology, you might have done a lab with another ciliate called tetrahymena. They usually do a tetrahymena lab in cell if you've taken that. Tintinnids are related to both of those. They're shaped more or less like a cone. Usually there's a firm outer covering. They look a little bit like vases or drinking glasses, except there's a cell sticking out of the open end, covered in multiple beating cilia, so the tintinnid can swim. Foraminifera, or what my major professor specialized in, the cell itself kind of looks like a very highly branching amoeba. So there's no really fixed shape, but there's a network of extensions from the cell called pseudopods. And there are some naked forams, but most of them build a shell-like covering around the main body of the cell that's called a test. Nothing to do with test as an exam. Latin for a pot, right? Like a little clay pot was a testa. And a very small clay pot was not a testa. If it was really small, it was a testiculus. So testicles means little clay pots. I guess because it's a very bad thing if you hit them and they break or something like that. I don't know. I don't want to think about it anymore, frankly. But yeah, test is sometimes used just to mean any kind of biological structure that looks like a pot if you're on drugs. Radiolaria are also amoeba-like cells with a network of pseudopods, but they have an internal skeleton made up of silica. Tintinnids, the outer covering is usually organic. Forams, it can be made of several things, but the most common is calcium carbonate. Radiolaria have internal skeletons almost always made up of silica, except for the one weird one that's made up of strontium sulfate, but never mind that guy. So here's a tintinnid. You can see the overall shape looks a little bit like a wine glass with no base. The glass is made of a tough, translucent protein, and the cell is inside where the wine would be if this were a glass. And at the open end, you should be able to see a bunch of little hairs. That's the cilia on the tintinnid that enables the tintinnid to swim and also directs food particles into a mouth structure called the cytosome, the broad end. Inside the tintinnid, you see those greenish circles. That's food vacuoles. That is whatever the tintinnid has eaten being digested. It's probably algae of some sort, maybe blue-green bacteria. And you may never see these unless you get to look at marine plankton, but they're extremely abundant in the oceans. They can be up to several thousand per liter, and they're critical as a link in oceanic food webs. They feed on small phytoplankton, so they will eat those blue-green bacteria and coccolithophorids and dinoflagellates and all of that. And in turn, they get eaten by small crustaceans and larval fish. And then those get eaten by bigger fish, and those by bigger fish, and so on. So tintinnids are an important link in the food web. They take that primary productivity, that fixed carbon that's being made by the phytoplankton, and then by eating it and getting eaten, they pass that up higher up in the food web. So ecologically very important, even though they're not the kind of thing that you're likely to see. There's a few relatives, but there aren't any real freshwater tintinnids. So you're not going to see these in Lake Conway unless things get really weird. Most forams are actually sit on the bottom. We say that they're benthic. But there's a sizable group of them that look like this. At the center of this picture, you see what looks like a little cluster of bubbles. That's the test. That is actually made of calcium carbonate in this case, and the main cell body is in there. And then extending out from the cell body through a bunch of tiny holes in the test are some very long pseudopods, extensions of the cell. And these can be withdrawn, new ones can be created. The pseudopods can sometimes join together and make a real net, a network. The pseudopods trap food particles, and the pseudopods also slow down the sinking rate and enable these guys to float in much the same way that the fuzz on a dandelion or a milkweed seed slows the sinking rate and enables those seeds to drift for quite a long distance. So there's a planktonic foram. Like the coccolithophorids, they'll live very abundantly in certain parts of the ocean. They'll feed, they'll reproduce, and they'll die, as all things must. And those tests sink to the bottom. And sizable portions of the deep ocean floor are covered in many feet of dead foram tests. This is a picture of a part of the deep ocean floor taken from a submersible, basically a small two-man submarine that can actually descend to, I want to say like 8,000 feet or something like that. That's not the very deepest parts of the ocean, but it's plenty deep. It's called the Alvin, and it's been used in research ever since at least the early 1970s. It's really cool. I actually knew somebody who had been on it. I've never gotten to go, but I knew someone who'd made some trips on the Alvin, and it's cool. Let me stop and tell you this. If you go on the Alvin, what they'll do is give you a styrofoam cup, and you can draw anything you want to on it with waterproof markers. And they'll take the styrofoam cup that you've decorated and carry it on the outside of the Alvin, I think in a little bag that they've got. Now think about this. Styrofoam is really light because it's filled with tiny air bubbles. If you take a styrofoam cup down 5,000 feet below the surface of the ocean, the pressure that you're under from 5,000 feet of water crushes the styrofoam, squeezes out all of the air bubbles, and when you come up, that styrofoam cup is hard as a rock and about the size of a shot glass. And of course, your design is now squished down too. It's kind of like if you ever played with Shrinky Dinks, it's kind of like that. So anyway, so yeah, I had a friend of mine who got to do that and had a little collection of little styrofoam shot glass cups. Some people will actually take down the styrofoam heads that wig sellers use and come back, and the head is shrunken down to a couple of inches in size. It's slightly disturbing. Anyway, the mud that you're looking at is all foram ooze. It's all tests of dead forams building up into deposits that may be many feet thick and that may themselves harden into deposits of soft rock, very much like the White Cliffs. And then radiolarians look like this. Like the forams, you can see that the basic build looks like a milkweed pod or a dandelion fluff. You have lots and lots of very thin extensions of the cell, the pseudopods extending out from the center. Most of the cytoplasm is inside. It's the dark zone in the middle, and that's where most of the organelles are. And then surrounding that, you've got a zone of very frothy cytoplasm that's mostly made up of vacuoles called colima. And outside of that, you have the zone with all of the little radiating pseudopods. Now, the cool thing is that there's a membrane structure between the internal endoplasm and the frothy ectoplasm. And that membrane structure is where the radiolarian makes this. They've got skeletons made out of silica. And yes, you can get areas on the deep ocean floor that are covered several feet in radiolarian ooze, just like you can areas with coccolith ooze and foram ooze. And these skeletons have this amazing and beautiful symmetry to them. They look like something you would put on your Christmas tree, and they're secreted inside the radiolarian cell right there on the capsule. So I'll leave you with this very pretty geometric object created by organisms in the deep ocean. And on Wednesday, we'll talk about animal life, and we'll find out what eats all of these critters, and talk a bit about food webs in the plankton. So I will go ahead and stop recording and